Okay, let us take a look at our next group of connective tissues, and we're going to start off here with compact bone. And we're looking at a nice cross-sectional view, which is different than what we would see in a longitudinal view. So that's another important factor to kind of point out here. We're looking at a nice section with India ink as our stain. We do have some H&E stains that we can use as well, and that's going to give us more of that pink purple coloration. And at a low magnification here, and this is our lowest mag, I have it toggled out, we have a nice bird's eye view of compact bone. And this is really, really distinct. It's very easy to identify, I feel, compact bone right away because of all of these basically miniature tree rings is what it looks like. So it looks like all these little trees and kind of cross section. And all of those ring structures are what we call osteons. And so compact bone is formed with the bone matrix, and our bony cells, our osteocytes, are making this bone matrix in these concentric rings, and those concentric rings form units, right? And these units are each called an osteon. We do also have interstitial, what we refer to as lamellae, and kind of this filler bone matrix that is kind of in between all of these tree units. If we had a nice peripheral view, and maybe this is a little bit here, we would also have what we refer to as circumferential lamellae, which is going to be kind of layers of this bone, not making the osteons, but have kind of a surface, right? Like a nice surface layer of the compact bone. So why don't we go ahead and zoom in and look at just like maybe one of these osteon units. You can see that there is a uniform pattern through all of them. They're not all exactly the same size either, which is kind of interesting to note. We can see this kind of central section. We have all these rings, and then we got all these little black dots all over the place. So let's take a look at our one osteon, and actually it zooms in quite nicely right where we're going. So if I were to circle out and put up kind of a peripheral border here, that would be the entire osteon. So everything that's within this kind of tree ring unit is an osteon. The central area here is what we refer to as the Haversian canal or the central canal. I use both terminologies. And this is where we would have like a vascular flow. All right. Each one of these tree rings, right, of this bony matrix is going to be referred to as lamellae. And you can think of it almost like laminate flooring, like you have layers and layers of this bony material. And it actually is kind of laid down when it actually is formed. It lays down in this kind of one tree ring after another after another. What we also see are these black dots that are here. And unfortunately, with how this is staining and because it's all the living tissue is washed out, the ink collects in the space where the cell would exist. So our osteocytes, and we have to use the term site here because it's a mature cell, because this is a mature bone, is going to exist in these little black areas. Okay, so what would be in here? So if this was living tissue and we could go up even higher in magnification, I don't know how far we can go here, we could see that there would be a space, there would be a cell, it would have a membrane and cytoplasm and a nucleus, but we don't see that here. We just kind of see where the cell would exist. And so within this area, you could say this is where the osteocyte would live. This is where we would see a lacunae or a space that is around that cell. And then what you also can see, and the stain kind of collects, are all these kind of like little hairline fractures, right? You can see all these little lines here, and those are all referred to as caniculi. And again, when you see that that suffix of uli, iole, ule, it really means kind of a smaller version of something else, right? And so these canals or these caniculi are little channels that lead from the vascular flow here to each of the lacunae where the cell would be housed, so that we can have molecular exchange. You have to think like all of this matrix. So all of this is bone matrix in here. It's not living, but the cells are living. And how do they stay alive when they're basically cemented in bone matrix? We can't just like cement them in, or they would die off. And so we have to be able to have some sort of molecular flow here, and these caniculi allow for those channels to exist. And then the cells themselves actually do have like cytoplasmic extensions that do extend into the caniculi, which is kind of amazing, really. So you can see how many living cells would be here existing in one little osteon, right? So quite a few that make up this entire osteon and kind of exist even within the lamellar layers. Out to the side, right? And so what is all of this here? Well, that's considered to be lamellae as well, or kind of layers of bone matrix. And it doesn't have a pattern like an osteon would, so we usually refer to that as interstitial lamellae, okay? And even if I zoom back out here, let's kind of go up to the top. Ooh, just take a moment. What do you think that is? That is going to be a little bit of a snippet of a Volkmann's or a lateral canal. Because remember, these are all blood vessels kind of traveling out of the screen towards you or going into the screen. And so we're looking down kind of like the top of the straw. We also have a very perpendicular system of these bigger canals, right? These channels that are going at 90-degree angles. And so every so often, depending on how the slide is cut, you'll be able to maybe see a little bit of that Volkmann's canal. And you can see a little bit more here too. So this is a cross-sectional view. So if we had one that was longitudinal, which you do have the ability to see that in your slide box, you would be able to see on that longitudinal view more of these Volkmann canals, although you would not see the osteons and kind of these concentric ring forms that are here. So up here at the top, if I kind of click in here, we can see, again, layers and layers of bony matrix in those lamellae. You can see, again, osteocytes all over the place. You can see some of the caniculi here as well. And so this would be more of that circumferential lamellae at the actual surface, kind of making a thicker layer of bone. And you would, if we had a bigger section of bone, you would see that this is actually can be a really, really large area as we get out to the surface. So something else we can do, we can always jump to the guide over here. Like here's an osteon and kind of showing you some generic ones. They're going to zoom into one and zoom into another one. You can see the central canal or the Haversian canal as they zoom in there. The lamellae, they're just kind of showing that in a different way. The actual like bony layers, I guess, is what they're trying to show you there. The lacunae, let's see where they're taking you. I wish we had arrows. It doesn't really matter. You can go to any one of these really. So this is just the space that would be around the cell. The caniculi, again, is just these hairline little fractures that are there. Let's see what else. Interstitial lamellae. Look, they're going to show you just again that inner space. Remnants of osteons. You can see it kind of is hazing out because other osteons are kind of coming into the area. You do remodel bone and you kind of replace things. So sometimes there can be older osteons within the area. And so again, you can absolutely go through this list over here and look to see what they have. There's other views that you can go to as well that is here. If I jump back to our guide, I picked this India ink version to take a look at. You can also use a different stain. Again, you can see some more longitudinal versus cross-sectional views. There is more of an H&E stain that is here, which I think is a little bit more difficult, but you get to see kind of more of a lateral versus, a cross-sectional view of like the diaphysis of a bone is essentially what that is. And so they're kind of demineralizing the bone in these different views. But the one that I think is going to be, more similar to what you guys are doing is here with that compact, that ground bone that is here, the compact bone. Okay, that's it for this particular slide.